I am Dr. Vandita Kanpal, Assistant Professor, Department of Zoology, Government PG College, Berinag, Uttarakhand. And my topic is Mendel's Laws of Inheritance, which is a topic of genetics of BSc second semester. Gregor John Mendel, who was the father of modern genetics. So he was an Austrian monk born in 1822 in Australia. So he was the first to demonstrate the mechanism of transmission of characters from one generation to the other. He also gave generalizations, some of which were later raised to the status of principles or called as laws of inheritance. And he constituted the foundations of genetics, so that he was called as the father of modern genetics. And he published his theory in Experiments on Plants Hybrids in 1866. But after giving his theory and published in work in recognized journals, his work was not recognized for about 34 years. So here are some reasons. Because limited circulation of the Proceeding of Brunn Natural Science Society, in which his work was published. Next reason was he could not convince himself about the conclusions being universal, since he failed to reproduce the status of Howitt undertaken on the suggestion of other scientists. So it was due to the non-availability of pure lines. Next reason was he was not aggressive in his personality. Other reason was the scientific world was being rocked at the time by Darwin's theory. That was a time when the Darwin's theory came in light and in the construct of evolution, and his work was published in Origin of Species in 1859. Next reason was the Mendel concept of stable, unblending and discrete units or factors for various traits did not find acceptance from the other scientists. There was no physical proof for the existence of factors or the material they were made of. So these are some reasons why his work non-recognized for about 34 years. But after his death in 1884, long before his work came to be recognized, it was in 1900 that three scientists independently rediscovered the principle of heredity already worked out by Mendel. The three scientists was Hugo de Vries from Holland, Karl Cornus from Germany, and Erich Tresmark for Australia. So he was known as the father of modern genetics because of its contributions in the genetics. So before learning about Mendel's laws of inheritance, firstly we learn about what is inheritance. So the inheritance means that every character is controlled by a gene that has at least two alleles. It is called monogenic inheritance. So study of inheritance of single gene means one pair of alleles of a character at a time, means monohybrid cross, is called inheritance of one genes. So basically the meaning of inheritance is the parents and offspring often share observable traits. So Mendelian inheritance is a set of primary trends which relates to the transmission of hereditary characters from one generation to another, or we can say that from parent to their offsprings. So it underlines much of the genetics. So inheritance is each organism has a large number of characters such as the height, skin color, flower color in plants, and each character is represented in an individual by two unit factors which are now called alleles. So the alleles occupy the same gene location on the two homologous chromosomes. When both the alleles represent the same gene, the condition is called homozygous, which we will learn in this slide, that about alleles and genes, which are called the units of inheritance. So alleles are one or two or more possible forms of a genes that are found in the same place, or we call it locus, same locus on the two homologous chromosomes. So when both the alleles represent the same genes, the condition is called homologous. Example, T and T, and a set of T means capital T and the small t. We will write it in the alphabets, capital and small. When two alleles represent different traits of same character in the individual having Such a pair of alleles is called heterozygous or hybrid. Means it is represented by a capital T and a small t. So there are two alleles. The dominant allele marks the expression of the another allele. Means the dominant overlap the recessive allele. So the dominant factor of allele is of a one pair of allele which can express itself whether present in homozygous or heterozygous state, and is the gene is the unit of inheritance which is carried from parents by a gamete. About genotype and phenotype. Firstly, we will discuss about genotype. It is a Greek word made up of two letters. Genus means the race and typus means the image, given by Jonassen in 1911. So it is the gene complement or the genetic constitution of an individual, which regard to one or more characters it possesses, which characters are irrespective of whether the genes are expressed or not. So for example, we can say that the genotype of a hybrid tall pea plant is capital T and small t, and pure tall plant, its genotype is capital T and capital T, both the T's are capital. And dwarf, the genotype of dwarf plant is small t and the small t. It is about genotype. Now the phenotype, it is also a Greek word made of two letters. Phenonym means to appear and typus means to imagine or image. It was also given by Jonassen in 1911. So it is the observable or measurable distinctive structural or functional characteristic of an individual with regards to one or its more characters, which is a result of gene products which are brought to the expression in a given environment. So the characteristic which appear in a phenotype may be visible to eye, like height of a plant, which we can see with our eyes, like height of a plant, color of a flower, or many other character. Special test for its identification, like serological test for blood group. So for recessive genes, the phenotype is similar to genotype, and for the dominant gene, the phenotypic expression can be due to its homozygous genotype or heterozygous genotype. For example, in a phenotype, we can state this example like phenotypic tall pea plant can be genotypically capital T or capital T or small t. We can write it like this. So genotype and phenotype is the two terms which is most important in the genetics of a gene on its homologous chromosomes, and it is not pure, and it is called hybrid for the character. So heterozygous does not breed true on self-fertilization. The example is we can write in heterozygous capital T and small t, and the homozygous with the capital T and the capital T. So these are about the homozygous and heterozygous. Here one question came in our mind that why he select only pea plant. So there are some advantages of selecting pea plant for his experiment. So here are some reasons, which are its pure varieties are available of pea plant, and it is a plant which showed easily detectable constructing characters, and flower structure of pea plant allow control breeding, and even plant is self-pollinated, but it can be bred can breed normally manually, and pea flower normally remains closed and it undergoes self-pollination. It is the reason why he selected pea plant, and it is an annual plant which have a short lifespan, and it give results early within 3 months, and it produce a large number of seeds per plant, and plant can grow easily and does not require after care so much care except only at the time of pollination. And its plants F1 hybrids we got, they are fertile. So here are some reasons why he selected the pea plant Pisum sativum for his experiments. Now one term is here Punnett square. So what is a Punnett square we will learn in this slide. So it was developed by a British geneticist. It was Punnett in 1927, and it is known after his name as Punnett square. So it is a checkerboard or square which is divided into two smaller squares as shown in the figure. I have shown this, and it is a graphical representation of to calculate the probability of all possible genotypes of offspring in a genetic cross. So the possible gametes are written on two sides, the top row, which is the horizontal row, and the left column, vertical column. Usually male gametes are written in top row and the female gametes in left column. So all the possible combinations are represented in boxes below in these squares. So from with this square we can get the various types of phenotypes and genotypes of any individual. Next is in this slide Mendel's 7p characteristics has been shown. So there are some characters, 7 characters, which is Mendel's taken, like flower color, flower position, seed color, seed shape, pod color, and stem length. And the traits are shown which are dominant or recessive, like purple for flower color and white recessive. Flower position is or it is axial or terminal. Seed color is yellow for dominant and the recessive for green. Seed shape is round for dominant and recessive is wrinkled. Pod shape and pod color green or yellow, or stem length or is tall or dwarf. So Mendel's 3 steps were involved in Mendel's experimental technique, that there was selection of pure parent plants and hybridization of pure plants for F1 generation and self breeding in hybrid plants for F2 and F3 generation. So he selected pure parent plants because he selected 34 varieties for his experiments and he allowed them to self breed for obtaining pure varieties. So the number were reduced to 22 and finally Mendel selected 7 pairs of pure or 2 breeding varieties of P as the starting material for his experiment. So at his starting material for his experiment he selected 7 pairs of pure true breeding varieties of P, and on self-pollination or self-breeding a pure variety gives rise to offspring have similar traits. Example like tall variety with tall offspring and a violet flowered variety with violet flowered offspring, etc. So all the characters of selected varieties had easily distinguishable alternate traits like tallness and dwarfness, violet flowers and white flowers. So Mendel satisfied himself as to true breeding nature of the variety through self-pollination. Any offspring not true to the form of the trait was eliminated. So two breeding plants were then used for the next step and they formed a parent, means the P generation. So these are some characters and constituting traits of Mendel's P plant. So in F1 generation, F1 results are shown. So F1 generation, all stamens from the flowers of the tall stem plants, means 100 to 200 centimeter height, were removed so that the tall stem plants acted as female plants. So all the pistils from the flower of the dwarf stem plants were also removed. So the dwarf stem plants acted as the male plants. So these plants were called parents, which are now expressed by the symbol P. And the cross pollination was done in which pollen grains from the dwarf plants were transferred to the stigma of the tall plants. So in then F1 generation, Mendel collected all seeds from in the flowers of the tall P and showed these seed in garden, and all plants that grew from these seeds had tall stems, and he marked these as plants of F1, means the first child generation, first offspring generation. And in F2 generation, Mendel allowed the plants for F1 generation to reproduce by self-pollination and the seeds were obtained. When he sowed these seeds, so these seeds grew into tall stem plants and dwarf stem plants of F2 generation, giving nearly a 3:1 of all stem and dwarf stem plants, which is shown in the this table. Most significant feature of this experiment was that dwarfnet disappeared in F1 generation but reappeared in about 25% plant of the F2 generation. So ratio is also given in the this table in Mendel's data. So about other different type of characters, like in seed shape and seed color, he got all round shape is all round in F1 generation and all yellow in F2 generation, and pot shape all full, pot color all green, flower color is all violet in F1 generation, and flower position is all axial in F1 generation, and stem height he got F1 results all tall, and F2 results are also shown, and the ratio is also given. Now we come to the main topic, which is Mendel's laws of inheritance. So Mendel has given 3 laws for his experiment, which is called as the Mendel's laws of inheritance, which is first is law of dominance, second law is law of segregation, and the third one is law of independent assortment. So at first we will learn about the law of dominance. So Mendel's postulate of dominance was raised to the status of law of dominance by Correns in 1901. So according to this law, characters are controlled by discrete or distinct units is called the factors, now which are called the alleles, and alleles occur in pairs, and out of 2 alleles only one allele expresses itself in the hybrid and prevents the expression of the other allele. So in other words, we can say that the out of 2 alleles only one allele expresses itself and the other one is remain recessive. So the one allele which expresses itself in the hybrid is called the dominant, and the other which is unable to express in the hybrid is termed as the recessive allele. So a capital letter is assigned to dominant allele. We will write capital T or any dominant allele, and the small t for tallness it is capital T, and the dwarfness is small t of a recessive allele. And Mendel experimented with Pisum sativum for 7 characters only. So in each case he found that one expression of or one trait of the character, like capital T or tallness in case of height, is dominant over the other expression of trait of the character, like small t or dwarfness in case of height. So this can be proved experimentally in this statement that we have taken 2 pea plants. Okay, one pure or homozygous tall, which goes height to the 100 to 120 centimeter, and the other pure or homozygous dwarf, means the less height, about 22 to 44 centimeter. Then we have caused the 2 and the raised their progeny called first filial or F1 generation. So all plants of F1 generation are tall, which is shown in this diagram. So all plants of F1 generation are tall. Height is goes 100 to 120 centimeter. So they have also received an allele for dwarfness. Okay, so that the allele for the dwarfness is present in the F1 plants can be tested by self-breeding them when individuals of F2 generation will be the both tall and dwarf in the ratio of 3:1. Therefore, in F1 plants, both the alleles for tallness and the dwarfness are present. But however, the allele for the dwarfness is unable to express itself in the presence of allele for tallness. Here the allele of tallness is a dominant and the allele of dwarfness is a called recessive. So first law of dominance we can see in this slide. It is a monohybrid cross showing law of dominance in pea plant. And what is the significance of this cross? So it explains why individuals of F1 generation express trait of only one parent. Second significance is law of dominance is able to explain the occurrence of 3:1 in F2 individuals. And the third one is it indicates why mixed population is superior as it hides many of the defective recessive alleles. So exception of to law of dominance, there are incomplete dominance and codominance are some 2 exceptions to law of dominance. The second law is called law of segregation, and this law has been called Mendel's first law by Correns, but it is the second law given by the Mendel. So it states that according to the law, this law, 2 alleles of a gene controlling each character stay together in the individual, but during gamete or spore formation by meiosis, the cell division, the alleles of a pair segregate, means separate from each other, so that a gamete or spore carried only one allele of a character at the time of segregation. Since one gamete or spore contains only one allele of a character, so all gametes or all the spores are always pure. So the law of segregation is also called the law of purity of gametes of spores. So this law is based on the fact that the alleles do not show any blending, means the mixing, and both the characters are recovered as such in a F2 generation. So one of these is not seen in F1 generation. So of course a homozygous parent produce similar gametes when a heterozygous parents produce two kinds of gamete, and both the having one allele with equal proportion. So the two important consequences of this law are each gamete or the spore contains only one allele of a gene, and the ratio obtained is 3:1 in the F2 generation of a monohybrid cross, and therefore the 3:1 ratio is commonly known as the monohybrid ratio. In this slide, we will explain about this law of segregation. So the law of segregation can be explained with the help of this monohybrid cross. Say between a pure tall pea plant and a dwarf pea plant. So the hybrids or plant of first F1 generation are all tall, although they have also received the allele for dwarfness. Even because they also receive the allele for dwarfness, but in the F1 generation all plants become tall. So it is because the allele for tallness is dominant, while the factor for dwarfness is recessive. So dominant over recessive. If the hybrids are allowed to self-bred, so the plants of the second generation or F2 generation appear to be both tall and dwarf in the phenotypic ratio, in the ratio of 3:1. So further, self-breeding of these plants shows that the dwarf plants breed true. Okay, that is, it means produce only one dwarf plant. Only dwarf plant they produce, not one. Only dwarf plants they produce. Amongst tall plants, 1/3 breed true, that is yield only tall plants, and the remaining 2/3 of the F2 tall plants are 50% of the total F2 plants. They behave as a hybrid plant and produce both tall as a dwarf plant, and the ratio is the same, 3:1. So therefore we can say that the F2 phenotypic ratio of 3:1 is genotypically 1 pure tall, ratio 2 hybrid tall, ratio 1 dwarf. So this cross show that even F1 plants show only one alternative or dominant trait of a character. It actually carries alleles of both the traits of the character, because it happens because the second alternative or the recessive trait appears in the F2 generation. So therefore F1 plants are genetically hybrid in the above case, where one is represented by capital T and other is small t. And the next is F1 plants are a product of fusion of male and female gametes. So as they carry the gene complement of capital T and small t, the fusing gametes, the gametes which fuse together must bring in only one allele each, means the capital T from both the capital T and the small t from both the small t parents. Next is in F2 generation is produced by self-breeding of the F1 plants. So F2 generation consists of three types of plant: pure tall, hybrid tall, and dwarf. So this is possible only when in some condition. First one is the two alleles present in the F1 plant segregate, means separate during gamete formation. Second condition may be gametes carry a single allele for a character, means 50% of one type and the 50% of the second type. And the third condition may be the alleles get distributed randomly in the offspring due to random or chance fusion of gametes during fertilization. So the conclusion is the homologous chromosome carry alleles. So there are two homologous chromosomes of each type which carry two alleles of each character. So the sexual reproduction requires meiosis for formation of gametes, and meiosis reduces the number of chromosomes to half, and a gamete carries only one chromosome of each type. So therefore only one of two alleles passed into a gamete. So 50% of the male and female gametes formed by the F1 plants, which possess the allele for tallness, while the remaining 50% carry the allele for dwarfness. So the law of segregation is most fundamental principle of heredity that has a universal application with no exception, And some workers like Batson call the law of segregation as the law of purity or gametes or the spores, because segregation of the two alleles of a trait results in gametes or spore receiving only one allele out of a pair. So as a result, gametes which are formed are always pure for a character. So there is a non-mixing of alleles. Now the third and last law of Mendel, which is called the law of independent assortment. So according to this law, the alleles of two pairs of traits separate independently of each other during gamete or spore formation and get randomly rearranged in the offspring at the time of fertilization and produce both parental and a new combination of traits. So in fact, there is a occurrence of two important events in this law. According to this law, first is separation of the alleles of two pairs of traits which are independently of each other during gamete or spore formation. First one is this. Second, there is a rearrangement of alleles, means random union of alleles in the offspring at the time of fertilization. So this law states that each allele of any one pair is free to combine with any allele from each of the remaining pairs during the formation of gametes or the spore formation. So this law can be explained with the help of this dihybrid cross. In this cross, we can see that a cross is made between a pure round yellow seeds pea plant, which is showed by capital RR and capital YY, with wrinkled green seeded pea plant, small RR and small YY, and yellow color is dominant over green and rounded seed shape or wrinkled seed shape. So F1 plants means the hybrid plants are all round and yellow seeded, and F1 plants are allowed to self breed and produce in F2 generation. And F2 generation has four types of plants. We can see that rounded yellow, rounded green, wrinkled yellow and wrinkled green in the ratio of 9:3:3:1, and each of the character is considered separately shows a ratio of 3:1 as found in a monohybrid cross. So seed color, we can see that yellow 9 plus 3 is equal to 12. Seed color, we can see green 3 plus 1 is equal to 4. So ratio between yellow and green is 12:4, or we can say that the 3:1. Next seed shape, if we see the round is 9 and 3, total 12, and the wrinkled 3 plus 1 is 4. So ratio between the round and wrinkled is 12:4 or 3:1. So the F2, the ratio of 9:3, 3:1 further shows two type of recombinations. We can see that the wrinkled yellow and rounded green. So they can be produced only if the alleles of two different characters are free to recombine. That means the separate and combined independently of each other. So the same can be confirmed with the help of Punnett square, which we have seen in earlier slides. So in this slide, hypothesis independent assortment is seen in the form of Punnett square. We can see in the P generation, parent generation, round yellow and wrinkled green, crossed and gamete are produced in F1 generation, round yellow, which is shown by capital R small r and capital Y small y, and the gametes are produced. And F1 generation, the phenotypic ratio is round yellow is 9, round green 3, wrinkled yellow 3, and the wrinkled green 1. Again, genotypic ratio is 1:2:2:4, 1:2 for round green, for wrinkled yellow 1:2, and the wrinkled green is ratio 1. So this is a result of a dihybrid cross when the two parents differed in two pairs of contrasting traits, seed color and seed shape. It is shown in this diagram. So now we come to the conclusions of Mendel's experiments. So after performing his experiments in garden pea plant Pisum sativum, he came to four important conclusions from his experiment. These are the first one is the inheritance of each trait is determined by units or the factors, which is now known as the genes, that are passed on to another generation and they are remain unchanged. Next is an individual inherits one such unit from each parent for each trait. And third one is a trait which is not meant shown up any individual But if we cannot see in any individual, but a trait can still passed on to the next generation from its parent. And the last conclusion is the genes for each trait segregate, means separate themselves during the gamete production. So these are the conclusion of Mendel's experiments. And at last I want to say thank you.